see this now. Is that okay?
Yeah, that's working great. Thanks, Colin.
Okay. So this talk is going to take 15 minutes to 20 minutes. I'm going to whiz through an awful lot of images that people have kindly provided me with. This is a really nice talk to give because it gives me the opportunity to share some of the best images and data of other people. So I haven't had to do all the work for this one. Let's just make a start. I'm going to start with a very brief history and give you a recent timeline of how the microscopy facilities have developed at the MBA. And then I'm going to focus mainly on optical imaging, talking about these aspects of optical imaging: confocal, two-photon light sheet, Mesolens, PAM microscopy, flow cam, and laser capture. And in the end, I'll say a little bit about electron microscopy. So microscopy's really been with the MBA from the very early days. This image at the top left here shows the very first microscope. We didn't actually use one of these because we weren't in existence when this one was made by Antony van Leeuwenhoek. So this is a microscope based on a tiny glass bead. But over the decades and centuries, these microscopes have evolved into what we have now. This is one of our most recent acquisitions at the bottom, and I'll tell you a little bit about that in a few minutes. I would also point out to you this microscope, this black one in the middle, which is possibly one of the earliest imaging microscopes. It's an old microscope that enabled the specimen on the stage to be projected onto a screen. And I understand there's an example of this still in the MBA archive. Okay, continuing with history, I just want to indicate one or two of the real microscopy pioneers at the MBA. And we can start with the very foundation of the MBA with Thomas Henry Huxley, a great microscopist and evolutionary biologist himself. One of the things he's famous for is his study of coccolithophores in chalk, and it's good to see that the MBA is certainly carrying on that tradition in research. Other great examples are Marie Lebor, who was a plankton biologist, and she created some of the very early images of phytoplankton and zooplankton, collected mainly around the English Channel off Plymouth. If we move on, another notable example is Mary Park, a world expert and renowned person on phytoplankton taxonomy and microscopy. She established the Plymouth Culture Collection, or was instrumental in bringing it up to the modern day. We mustn't forget Hodgkin and Huxley, who they weren't members of staff of the MBA, but they did a lot of their work at the MBA on the squid giant axon. They were some of the first people to develop electrophysiology along with microscopy, and of course they won the Nobel Prize for their work on the nerve impulse and the theory of ion channels. So the last three people I want to introduce together really have brought the MBA right into the modern microscopy era. And Anne Warner was instrumental in this. Anne Warner was a professor in University College London, and she initiated the microelectrodes workshop that we still hold every year to the present day. Anne Warner provided huge advice and expertise on modern microscopy and modern electrophysiology. Similarly, David Ogden, who really carried the flag for that workshop and still does, has expertise in biophysics, particularly patch clamp electrophysiology, and again, coupling that with microscopy. And David really brought Brad Amos into the MBA environment. David persuaded Braz to start a microscopy course at the MBA. Brad Amos is a very famous microscopist and inventor. He actually was instrumental in inventing the confocal microscope. He invented the confocal scan head. And of course, he also invented the Mesolens, and we have examples of both of them here at the MBA. So this is just a recent MBA microscopy timeline. I'm not going to go into any detail here. If we go back to the 1970s, we established, mainly through the work of Quentin Bourne, an electron microscopy suite. And then during the 1980s, there was an expansion of microscopy and electrophysiology. And around about the mid-80s to late 80s, we started to do the first quantitative fluorescence imaging and photometry. And we acquired our first confocal microscope, again, in the late 80s, early 90s. That was from through a BBSRC grant. What I'm really going to focus on is the last 20 or so years. We acquired another confocal microscope after that, and then a Zeiss LSM510 microscope, which we still use today. And so over the past 2 decades or so, we've seen an increasing population of our microscopy facility, including these microscopes. I'm not going to go into any detail in this slide. I'm going to talk about them all in over the next few minutes. I'll also mention the FlowCam microscopy, which, of course, the continuous plankton recorder has brought to the MBA. And I'll say a few words about that at the very end. Okay, so much of my talk is going to concern fluorescence imaging, or optical fluorescence imaging. And we consider fluorescence to be the absorption of light through an excitation beam and the emission of light at a longer wavelength. So if we have a fluorescence molecule or a fluorescent substance, we can shine, for example, blue light on it. And if it's fluorescent, it might give off green light or red light or light of a longer wavelength. So that's the whole essence upon which fluorescence microscopy is based. So clearly, if you have fluorescent molecules in a cell, you can use fluorescence microscopy to selectively excite those molecules and then selectively image them in a microscope. And down on the bottom left here, there's a very simple diagram of how a fluorescence microscope works. The excitation light comes in through a port in the microscope. It's reflected down to the specimen where it's focused by an objective lens. And then the emission light, which I've represented by a green rays here, passes through this special mirror. We call it a dichroic mirror, and that mirror reflects short wavelength lights and lets longer wavelength light pass through. So the longer wavelength emission from the specimen can pass through the mirror and be focused by the microscope eyepiece, for example, on your eye or on a camera. And down in the bottom right, we have a typical example of what we call a wide field epifluorescence image. And with fluorescence, we can use, for example, fluorescent dyes, for example, for DNA, cytoskeleton, for measuring calcium or pH within a cell. But more recently, we've turned our attention to using genetic reporters. We also call them biosensors. These are, for example, green fluorescent protein tagged proteins, calcium reporters, pH reporters, and reactive oxygen species reporters. And I'll just show 1 or 2 examples of them in a few minutes. So our workhorse fluorescence microscope is this Leica epifluorescence microscope. It's a very, very good microscope. It's equipped with temperature control on the stage. So that means we can do short and long-term time-lapse experiments. It's equipped with a perfusion system, so you can change the medium around the cells. In particular, it's equipped with a very fast, ultra-sensitive CMOS camera. This enables very low fluorescent signals to be measured with millisecond time resolution. And it's also equipped with an LED array for multi-wavelength fluorescence excitation, which gives it a very broad application. So I'm going to just show you 1 or 2 examples of how we use this microscope. And I'll jump straight in with this one. So this is an image of Phaeodactylum, diatom cells. expressing a genetically encoded reporter for calcium, and we call that reporter R-gecko. I'm just going to run this short video, and the colour in the cells will change as I show the video, and that's in response to giving the cells a slight osmotic treatment. So we go from seawater to dilute seawater. And we're measuring the very, very low concentrations of calcium in the cytosol in these cells with this reporter. So if I just run the video, this is more or less in real time. We could see all the cells flash there, and as we give successive osmotic treatments, the cells all begin to flash with their own particular frequency. I'll just play that again in case you missed it. So this is allowing us to measure sub-micromolar levels of calcium concentration within the cytosol in the cells. And this is the work of, this slide is the work of Catherine Helliwell and Friedrich Kleiner. We can also, for example, measure reactive oxygen species within cells. Again, we use a genetically encoded reporter. This one's called RhoGFP2 OP1. And at the bottom, we've got some phaidactylum cells expressing that reporter. And some of these reporters have a very useful property in that their spectrum shifts when they either bind an ion or they become oxidized or reduced. In the case of this reactive oxygen reporter, the excitation spectrum of the reporter shifts when it's in a reduced state from the oxidized state. Now, without going into any detail, that allows us to carry out ratiometric quantification of actual levels of reactive oxygen in these cells. And on the right, I've just showed two examples of Jack Dickinson's PhD work, where he's used this reporter, again in phaidactylum cells, to measure changes in reactive oxygen within the cells in response to high light conditions. Again, I'm not going to go into any details. I'm more than happy to discuss aspects of this work with people anytime they like. Okay, so that was epifluorescence microscopy. If you look at the bottom of this screen, you can see, I showed you this image already. This is a wide field epifluorescence image that we'd get from a normal microscope, this one on the left. But you can see it's quite blurred. That's because there's a lot of out of focus information. Again, if you just look at this ray diagram, we can see how that happens. Here's the blue excitation light going in, the blue arrows. They are focused into the specimen. But of course, the light doesn't just focus to one spot in a normal microscope. It goes through the specimen. On the left here, we can see it goes through the specimen as a cone of light. It's focused to a spot, but above that spot and below it, there's light going through the specimen, which creates out of focus information. And I've represented that by this black arrow here. And that out of focus information will come up to the camera or the eyepiece or your eye and appear as out of focus blur. Now, a very special trick can be employed using a confocal microscope to get rid of that out of focus information, and it's a very simple trick. And it simply involves putting a pinhole in front of a detector so that all of that out of focus information is excluded from the detector. But of course, that also means that you only see a very, very small part of your image. So the other special trick that a confocal microscope uses is that rather than using LED or normal light to illuminate the specimen or excite the fluorescence, it uses a laser which is scanned to and fro across the specimen in a raster formation, just like an old television scan would make a television, an image on a television screen. So that point is then scanned through the specimen, and the light emitted from that point scanning by the laser is focused on the pinhole, so that the detector only sees light which is coming from this confocal plane in the specimen. And on the right we can see a confocal image of the same cell you can see on the left, but here all the out of focus information has been removed by the laser scanning and the insertion of the pinhole in front of the detector. And of course the signal from the detector is synchronized with the scanning of the laser, so that the computer allows us to build up an image of what we call an optical section through the cell, because that's a section just through one plane in the cell. And these images were provided by Glenn Wheeler. So I'm going to show you some examples of confocal microscopy. First of all, these are going to be images from our Zeiss LSM510 confocal multiphoton microscope. We've had this microscope now for almost 18 years, so it's been a real great workhorse for the lab. It has a multi-laser array and scanner unit, so it can excite several different kinds of dyes. It has a spectral analyzer, so we can look at the emission at different wavelengths. But it also has a pulsed infrared laser, which allows us to do what we call multiphoton imaging. And I'll say a little bit about that in a second. So these are just some examples of how we can use a laser scanning confocal microscope. So on the top here we have some examples of a haptophyte alga, and this is stained with a dye which can allow us to image the silica scales that it produces, and that's what the green fluorescence is. And the red fluorescence at the bottom of the cell is the chlorophyll in the chloroplast. So on the left we have a representative single confocal section through a single cell. But we can also take what we call a Z-stack, which is a series of sections vertically through the whole cell. And then we can project all those sections together in one plane and get what we can see on the right, which is a Z-stack projection, which allows us to see more detail all through the cell. So in this case, we can still see the silica scales, we can see the chlorophyll in the chloroplasts, and we can also now capture the haptonema, which is this line coming out of the bottom of the cell here on the left. And on at the bottom here, we've also got an example of a stained polysaccharide from a coccolithophore, Coccolithus browrudii. And we've used a special dye to stain a particular polysaccharide on the surface of this cell. On the left, we've got a single optical section, and on the right, we've got a Z-stack projection, which shows the very interesting pattern of polysaccharides arranged around the surface of the cell. We can also use confocal microscopy to monitor ion concentrations, and I'm just going to, within cells, and this is just an example of how we've used confocal microscopy to look at intracellular pH, this time in a diatom. And on the top, we've used a fluorescent pH indicator, which we call SNARF. And this basically changes color when the pH changes. So you can see the pH, two different pHs within that cell after a particular experimental treatment. But we can also use genetically encoded pH biosensors, such as fluorine on the bottom here. So we've got cells of the diatom Phaeodactylum, which are expressing their own pH biosensor, fluorine, which is what these two fluorescent images are on the left and the right here. And using the information from these images, we can plot in experiments changes in pH within the cytosol of the cell. And this is work of Dorothy Kottmeyer. I'm going to show you another example of how we can use Z-stack imaging and with confocal microscopy to get some really neat imaging. This is a time-lapse sequence of 3D images taken by Davis London of a chytrid fungus as it develops from a single spore. So this is a single spore in the middle. I'm just going to run this video It's quite fast, so don't blink. Oh, sorry, not too fast. So this is a time-lapse image of a chytrid fungus which develops into a hyphal network and then suddenly sporulates at the end. And we can use sophisticated image analysis packages, of which we have several types. And I'm just showing you an example here, again from the kind of imaging that I showed you in the previous video. Again, this is by Davis and Michael Cunliffe, and they've used a rather complex imaging analysis protocol to identify the different patterns of spore development, again in a chytrid fungus. So on the right here, I've just listed some of the image analysis packages that we use in analysing all these different kinds of images. I'll just point out the new package that we have, which is MRIS. It's very comprehensive, very powerful image analysis package. We've only just acquired that. So this is for everyone to use, and please come and talk to us about it if you want to do some image analysis with this new package. Okay, I'm going to move on now quickly to another kind of confocal microscopy, but this is two-photon microscopy. A two-photon microscopy, very briefly, uses a very high intensity femtosecond pulsed infrared laser. The laser develops pulses of infrared light in very, very short bursts, femtosecond bursts. And this laser is, again, focused through the microscope, focused by the objective into a tight focal spot. And within that spot, there are so many photons that the probability of a molecule being hit simultaneously by two photons rather than just one photon, which is what happens in normal fluorescence, the probability becomes very significant. So we can have simultaneous excitation of fluorescent molecules with two photons. That effectively doubles the energy packet to the molecules and effectively halves the wavelength. So from infrared light, we can get blue or ultraviolet light, but only at this focal point. That means that there's no out of focus information. It means that we have reduced scatter because we're using infrared light to go through the specimen, which isn't scattered very much. And it means that we can get better depth penetration of thicker samples. I'm going to show you an image of an example of the use of two-photon microscopy in the lab. And this was taken by Venki Modapalli here in the MBA. It's in a sequence of optical sections from Nematostella, an anemone at the planula stage. And the video just shows a sequence of stacks taken through this specimen. And I'll just show it now. So the blue represents the nuclei, yellow represents tubulin, and red or pink represents ecahedron, which is labeling the walls of the cells. So this is just sectioning through this planula. We go down through it. From top to bottom or bottom to top, I'm not sure what the orientation is here. So it's just a good example of how two-photon microscopy can allow us to use effectively ultraviolet light to label the nuclei here and to effectively probe quite deep within the specimen. Okay, so our most recent acquisition is a Leica SP8 confocal, which does everything I've just shown you the previous confocal microscope does, but actually does more. But it's also equipped with digital light sheet capability. So what is digital light sheet capability? So if you just look on the top right diagram here, On the left, we have an example of how we would image something using confocal microscopy by scanning a spot through a specimen back and forwards. Of course, what we do get then is a lot of photo damage because we're shining an awful lot of light on this specimen. On the bottom, this is an alternative way of illuminating an optical section within a specimen, and that is to make a sheet of light and pass that horizontally through the specimen. And then we'll only get photo damage, if any, through that very, very small section of the cell. To illustrate that, I'm just going to show you a short video. It's not one we made. It's a video from the manufacturers of this microscope, Leica, and it illustrates how light sheet microscopy actually works. So I'm not going to say anything much during this. Just watch this video and it's a good graphic representation of how light sheet microscope actually works. Okay, I hope you got that. That was quite clear. And I'm just showing you one or two examples of some preliminary light sheet imaging that Ben Wheeler's carried out here at the MBA during one of our microscopy workshops. So on the left image here, we have a single light sheet image of a large diatom, Odontella, and we've stained some of the vesicles within the cell with a fluorescent dye. And on the right, we have a time-lapse light sheet image of I think these are the chloroplasts moving around inside. They've stopped. Let's start them again. Yeah. Anyway, you saw that anyway. The chloroplasts are moving around inside the cell. So light sheet imaging does allow us to do long-term time-lapse imaging without very much photo damage at all. And another advantage of light sheet imaging is that it reduces photo bleaching of the dye. And because we are illuminating a larger area than a single spot scanning on a confocal microscope, it allows us to image much faster than confocal microscopy. So it's very good for physiological imaging. Okay, so now I'm just going to show you another image that we've acquired more recently from our new Leica confocal digital light sheet microscope. This is actually a confocal microscope image, but it just shows you some of the imaging capability of this platform. This is another one of Venke's images. It's of a calcareous sponge, and it's a confocal three-dimensional projection. So Venke took confocal sections all the way through this specimen from top to bottom, and then the software on the microscope reconstructed a three-dimensional image of the whole organism. So I'm just going to run that video now. This just illustrates some of the imaging power of this system. And now we're just seeing a sequential display of the stacks as he took them through the specimen. You can see the white lines there are all the cilia, which are labelled with a fluorescent tubulin stain. This is another image acquired with the same microscope. This is an image taken by Davis. The red is chlorophyll, and that's a large diatom. That's all the chloroplasts inside a large diatom. But you can see it's surrounded by very small bacteria. So Davis actually imaging what we call the phycosphere in this organism. And in the blue, we have the DNA stain of the nuclei. So using a DNA stain here, we can image the bacteria. And by looking at the natural chlorophyll fluorescence, we can image the chloroplasts within this large diatom. I just mentioned that this image has been shortlisted for the British Phycological Society Hilda Cantor Lund image competition. So fingers crossed with that one. Okay, I'm going to whiz through some more aspects of the different kind of microscopes we have. I'm going to start by mentioning TIRF microscopy. TIRF stands for Total Internal Reflection Fluorescence. So what is that? Well, I've spoken until now about standard epifluorescence, where we have an incident excitation light that excites fluorescence within the specimen, and then we measure fluorescence emitted from that. TIRF relies on the phenomenon called total internal reflection. And if you remember your O-level or your GCSE physics or your A-level physics, you will know that if you shine light within a glass, say within a glass fibre, and if the angle of incident light on the surface of that glass exceeds what we call a critical angle, it will be totally internally reflected. Of course, that's the whole basis of fibre optic communication. Well, one thing that you probably didn't realise was that when this happens, you get also what's called an evanescent field of photons on the other side of the glass. So this below we have inside the glass, for example, inside a cover slip, and above we have an evanescent field of photons which have, you might say, escaped from this totally internally reflected light. But that evanescent field is very, very narrow. It's less than 100 nanometers. So if you have anything which is fluorescent within that evanescent field, you will excite fluorescence only within that 100 nanometers above the cover slip. Well, that can be very useful for looking at certain things. It's not useful for looking at deep within cells, but it can be very useful for looking at very small structures which have a tendency to adhere to glass. And the example I'm going to give you here is work from Glenn's lab again, where we've used this approach to image the flagella of an alga. The alga in this case is Chlamydomonas. And we can either load this alga with fluorescent dyes, or we can get it to make its own fluorescent reporter proteins. On the right here is just an example of how TIRF can be used in this specimen to specifically image the flagella. So at the top we have a wide field image showing the very bright fluorescence from the cell body. You can just about see the flagella sticking out. But if we turn the microscope to TIRF mode, we can image just the flagella and get rid of all that fluorescence from the cell body. So we've used this approach to image calcium within the flagella using specifically targeted reporters for calcium in the flagella or calcium indicators. I'll just show a video which shows calcium pulses within flagella. And these are flagella of single cells sticking down on the cover slip. You can't see the cell bodies, but you can see the flagella. And Glenn and ourselves have been interested in looking at how these calcium signals regulate the transport of molecules up and down the flagellum. So the flagella they grow in length by transporting proteins from the base to the tip, for example. And they also use protein transport to signal what's going on from the flagella into the cell body. And this bottom left-hand video shows the movement of intraflagellar transport particles, which have been labeled with a fluorescent reporter protein. And in the middle here, I'll show this video, which is a simultaneous time-lapse image of calcium, which is the left-hand streak there, and intraflagellar transport, or intraflagellar transport particles, or what we call IFT particles. I'm just going to play this image and look on the left. You'll see a flash of calcium in a second, and that flash causes those IFT particles to move away from the tip of the flagella and back towards the base. And then it all goes quiet with respect to calcium. The particles start to come back again, and then there's another flash of calcium, and the particles all start to move away again. So from this kind of experiment, and Glenn and his postdoc Cecile, and before her Peter Collinridge, have done an awful lot of work analysing how calcium is involved in regulating the transport of proteins within flagella. Okay, I'm going to move on now to the Mesolens. I think most of you have heard about the Mesolens. So this is a tool for high-resolution imaging of large numbers of cells. It has, so some of its key features are a very high-resolution, large array CCD or CMOS camera. The one I'm showing you here has 13,000 by 20,000 pixels. That's a very large array camera. It has a motorized stage, so we can put on perfusion, laminar flow perfusion and control temperature. We can do Z sections with it, as we can with a confocal microscope. But the key feature is this very large, low magnification objective. This is a ×4 objective. But if you know anything about objectives, you'll see that this has a very high numerical aperture. It's 0.5. This allows us to give aberration-free, very wide field images. And of course, it has all the features of a normal multi-wavelength fluorescence microscope. I'm just going to show you some examples of what the Mesolens can actually do. If we took a wide field image of, say, approximately 3 by 4 millimeters with a ×4 objective on a normal microscope, this is a bright field image, you would see something like this. This is a culture of our diatom phaidactylum taken with a ×4 objective. If we zoomed that up 10 times, equivalent to looking at it with a ×40 objective, we would see what we can see on the right here, which is not much more than we can see on the left. So we can increase the zoom, but we don't get any better resolution. We're limited in resolution by a ×4 objective. So on the bottom now, I'm going to play a video of something we've done very similar using the Mesolens. So we start with a ×4 image, non-zoomed, very wide field. This is about 4 by 3 millimeters in size. And then I'm just going to play this video which zooms up, and hopefully you can see the advantage of a Mesolens compared to a normal objective. So here we're zooming into the specimen, and what you can see are now the single cells of every single phaidactylum in this culture. And we can do the same with fluorescence. Now I'm zooming back out, switching to fluorescence mode. So with the Mesolens, we can look at tens of thousands of cells, but we can get single cell resolution as we zoom in digitally. And this image sequence was acquired by Serena Flory. So just some more Mesolens images, just to illustrate what it can actually do. This is just an example of a rock biofilm showing some diatoms. These are chlorophyll fluorescence images of diatoms. So at the top right hand, we've got the wide field fluorescence image taken with a Mesolens. You can see there's an awful lot of diatoms of different kinds in there. And then the bottom here, I've just zoomed this blue square, this very small area, and you can see the incredible detail that the Mesolens can retain in its images, which can be zoomed up for much more detailed analysis, right down to the single cell level. And this was obtained by Deirdre McLaughlin. This is another example just of an L4 plankton sample, which we acquired last summer. It just shows the potential for looking at natural samples of many different cell types. Again, this is a chlorophyll image of an L4 plankton sample. And again, this is another image taken by Davis. It's of a Chytrid fungus growing on a whole Daphnia organism. And this just illustrates the power of the Mesolens to selectively zoom in on particular areas of a very wide field image. So I'm just going to play this video now, and you'll see what I mean. Whoops, sorry. Here we go. So zooming right in onto single cells of the Chytrid fungus. And you can see how the scale bar's changing at the bottom. Okay, moving on. We not only do imaging, but we combine imaging with electrophysiology. We've used electrophysiology with microscopy in different combinations over a number of years. Just two examples here. I'm not going to show you any data. You'll be pleased to hear. We can do patch clamp electrophysiology, or we can use, for example, ion selective electrodes. We can measure things like pH, carbonate at the surface of a cell, for example. This is the work of Abdul Kratri. And over the years, this has allowed us to discover new ion channels and elucidate transport mechanisms for inorganic carbon in phytoplankton. Okay, another kind of microscopy that we use more routinely now is called pulse amplitude modulated microscopy. And this is a way of imaging photosynthetic efficiency in single cells or tissues of plants or algae. Without going into any detail at all, I will just say that when chlorophyll absorbs light, for example, blue light, that light is converted into electrons, which pass down an electron transport chain, which eventually creates chemical energy, which is used to fix carbon in photosynthesis. Now, when chlorophyll absorbs excitation energy, it can either be used to transport electrons, or it can be used as fluorescence. So the energy can escape as fluorescence, or it can be used to transport electrons. And if there are any defects in the electron transport chain, if the cells are not photosynthesizing very efficiently, then that will be reflected by increased fluorescence from the chlorophyll. So pulse amplitude modulated fluorescence imaging and fluorimetry is a very sophisticated way of measuring changes in chlorophyll fluorescence, which we can relate to the electron transport processes in photosynthesis. So the top here shows the PAM microscope that we have in the lab. It's equipped with a very nice temperature control system. And just below that, I've shown you some examples of a coccolithophore. On the left, a bright field, a calcite image, a chlorophyll image, and D is the image of photosynthetic efficiency, which we also call FV over FM. I'm happy to talk to anyone who wants to know more about pulse amplitude modulated imaging. Well, what we wanted to do is take this to sea to measure photosynthetic efficiency of single phytoplankton cells in a natural population. And where better to do that than the Southern Ocean? As you can see, the Southern Ocean presents some particular problems for microscopy. Pitching and rolling of the ship being the main one. So it really is only feasible to do this when the sea's quite calm, at least at the present time. Nevertheless, on a Southern Ocean cruise earlier this year, we were able to monitor photosynthetic efficiency of single coccolithophore cells, and that's what the bottom sections here show. I'm not going to go into any details. Here's a bright field image on the left, and on the right, the photosynthetic efficiency images. The calcite in those single cells is arrow-headed on the bottom left here, so we can say which of these cells were coccolithophores and which weren't. And then the table here gives the values of FB over M for all of those cells that we were able to measure. And we can combine that kind of study with PAM fluorometric image measurements over much wider areas and with depth profiles over whole regions of the ocean. So that's work in progress, and there's a heck of a lot of analysis still to be done. Okay, whilst we're on about natural populations, I will mention the CPR FlowCam. The FlowCam is a high throughput and analytical platform for analysis of plankton samples, and it's greatly extended the analysis of natural plankton populations. If you want to know more about that, speak to the CPR people, and I'm sure they'll be very happy to tell you, but it is a microscope and it is part of our microscopy platform. Okay, coming soon. We've recently acquired, but it hasn't yet been delivered, a laser capture microdissection microscope made by Zeiss. This allows the isolation of single cells from populations or cells within tissues, and it does that by using a laser to cut around the cell, and it then uses that laser to blast the cell into an Eppendorf tube, where that single cell or group of cells can then be analysed, for example, by PCR or any kind of molecular analysis. So we can do genetic analysis of single cells within natural populations or within cultures using this new approach. So it's not delivered yet. We'll hear a lot more about this, I hope, in the very near future. I'm not going to leave without mentioning the electron microscopy that we also do. We have a long-standing arrangement with Plymouth University, which allows us to use their EM facility. They have SEM, elemental analysis, transmission electron microscopy, and a focused ion beam SEM. And I've just illustrated that with some examples of our MBA work. On the left, some of Gerald Langer's coccolithophore images, and on the right, one of Davis's beautiful reconstructions of a koala flagellate. Okay, just to mention how this work was funded, a lot of the capital equipment was bought through BBSRC grants and an ERC grant, and more recently a NERC grant. The MBA, of course, has contributed significantly by providing matching funds, and of course I mustn't forget to mention that we now have a brand new microscopy suite down in the basement. It's not yet fully commissioned, but we are starting to move things down there, so look forward to seeing you down there in the near future. And with that, I'm going to finish. So thank you for listening.